Hello everyone, and thank you for attending today's webinar, An End-to-End Workflow for Exploring Spatial Biology through Multiplex Immunohistochemistry. I'm Beatrice Bowlby, Digital Editor at Biotechniques, and I'll be your host for today's event. Before I introduce you to our speakers, I'd like to quickly cover a few housekeeping items. You've joined the presentation listening via your computer's audio by default. If you would prefer to connect to a different speaker, you can adjust your audio settings by clicking the arrow next to the microphone icon at the bottom of the Zoom window. You will have the opportunity to submit text questions to today's presenters by typing your questions into the Q&A tab at the bottom of your screen. You may send in your questions at any time during the presentation. We'll collect these and address them during the live Q&A session at the end of today's webinar. Additionally, please let us know your thoughts on today's webinar by tweeting us at mybiotechniques. So, we are delighted to have Danielle Fales, Spatial Biology Manager at Bethel Laboratories, and Mark Zadie, Co-founder and Chief Technology Officer at Pathomics.io, presenting for us today. Thank you again for joining us, everyone. I'll now hand over to Danielle to start today's presentation.
Hi everyone, I'm Danielle Fales. I'm the Spatial Biology Manager here at Bethel Laboratories, a Fortis Life Sciences company, and I'm here to walk through the front-end operations of the workflow for exploring spatial biology through multiplex IHC. So a little bit about Bethel Laboratories. We're located in Montgomery, Texas, about 1 hour north of Houston, and we've been in the antibody manufacturing business for several decades. In addition to antibody development, all of our validation work is done in-house through a variety of assays, ensuring that customers get quality products. So we take each of our antibodies through IP, Western blot, and IHC, and then we also evaluate them in other applications such as ELISA, flow cytometry, and then multiplex immunofluorescence. For validation, a multiple assay approach is taken, which falls in line with the International Working Group for Antibody Validation's recommendations. They recommend at least 5 pillars for validation, and we've developed our own 6 in-house antibody validation pillars. These processes are extremely important as spatial biology workflows depend upon antibodies that are specific to the target of interest and also clean in the respect that they don't have a lot of non-specific staining or background. The output of your assay, whatever it may be, is always dependent upon the quality of the antibodies that you use, both primary and secondary antibodies. So you want to be sure that you invest the amount of time necessary in validating each of these prior to putting them in your assay. As I mentioned previously, we do everything in-house. For IHC and multiplex studies, this includes slide preparation. So from a histology standpoint, we're going to walk through our own in-house way, beginning with the first step, tissue processing, embedding, and microarray construction. So our technicians process and embed every cell or tissue necessary for the creation of our own microarrays. Once the blocks are created, then a separate technician does the sectioning and slide preparation, and then these slides are cataloged for future use. We also have several instruments that can produce whole slide images, but from a bright field perspective, we utilize the 3D Histech in-house. And by using these instruments for scanning, we standardize the results and just make sharing data much faster and simpler. So to the right here, there's a microarray of about 70 to 80 cores, and the resolution of these arrays when you zoom in on the actual image is quite nice. For multiplex analyses, we use a variety of instruments as well, but today's case study will showcase a workflow designed using the Lunaphore Comet. There are several instruments on the market today that utilize a variety of staining methods, but this instrument uses a fluidics approach. A slide is inserted into the machine, and then antibody flows across the slide under a sealed chip. The methodology can be observed in the diagram to the right, where primary antibodies are placed onto the tissue, followed by a fluorescently labeled secondary, stained with DAPI, and then imaged and eluted from the sample. This process can be repeated up to 20 cycles, preserving the tissue for use in later applications, such as an H&E stain, additional chromogenic stains, maybe some RNA work, etc. So when we initiate a project in-house, we always want to start with the specific research goals at hand. So we want to get a good feel for the number of markers that someone is interested in evaluating, because each instrument has sort of top end to the number of markers they can evaluate at a given time. So we begin with that, and then we look into acquiring those markers. We always recommend to begin with our in-house antibodies, but occasionally a researcher has a preference or history using certain clones, and or we run into the issue of not having a specific marker in our catalog at the time. When we want to look at obtaining antibodies, we get them based on a number of factors. Probably the most important is that we want to focus on the specificity of the target and then also the assay compatibility. And what we mean by that is we want to make sure that the antibody is listed as validated for IHC in this purpose. So for our case study of CD20, we would want to pull the antibody and its data sheet and make sure that there are specs listed and that these specs meet whatever standards we need for the assay. So in this case, we can see that it's validated for IHC as well as multiplex immunofluorescence, and that it's recommended for FFPE tissue, which is what we often use in the lab. CD20 here is one of our in-house clones, and you'll notice that this is a mouse host and that pairing requirements are listed. This isn't a necessary component for every single assay, but for the COMET, it's crucial to pair as many as you can, double host pair, so a mouse and a rabbit or a rabbit and a goat, so that you can combine your antibodies and then you can increase the number of targets you can evaluate at one time. The second step is looking at tissues that the antibody should be positive and negative in. So we want to pull tissue microarrays for internal validation testing that meet these requirements. We subject each of the antibodies to DAB chromogenic testing, and we do this using a standardized in-house protocol, which you can observe there. Upon completion of this staining, whole slide images are taken, and we evaluate and make sure that the antibody that we just tested was specific. In this case, CD20 would be expected on tonsil and negative on liver SCC. This meets the requirements, so we would take this antibody and we would move forward with it in fluorescent testing. The third step on this workflow would be to take the original concentration tested with DAB chromogen, and we would use this beginning concentration and we would build a sort of dilution series for the COMET instrument. And by this, I mean that if CD20 had a very strong signal at 5 micrograms per mil working concentration, our series that we would test on the Lunaphore Comet would include 8, 5, and 1. So we would want to include one higher and one lower than the original testing, just to ensure that any potential loss of signal by removing that HRP polymer secondary or amplification agent is recovered. So we would use the 8 in this case. If it's a very, very strong target, Oftentimes, we can use a little bit less and obtain the signal that we're looking for. So we want to include a 1 here. And then we always test, the original concentration. And so for here, it would be 5 micrograms per mil. Once these values are set, the tissue can be loaded onto the instrument. And you want to be sure that your tissue, whether it's whole tissue or microarray, is placed appropriately on the slide. For the comet, you want to ensure that it's fairly center on all sides, as the window for staining can be shown there in red. Then you load the tissue slide onto the platform and seal it with the chip, and your protocol can be assigned. The protocol here won't be going into too much detail on that, but essentially it's just set up to stain each concentration and then elute it off of the sample so that you have a good side-by-side and you can see how your various dilutions appear next to each other. So this is a good example of that, where we have dilution A, B, and C side by side on the same tissue, locked into the same area on the tissue. We also have these same exposure settings so that you can truly evaluate them side by side. So when you're trying to pick a dilution for an antibody moving forward, you don't necessarily want an antibody that has the histogram all the way to the right, this little bar here, because it doesn't leave a lot of wiggle room for downstream analysis operations. Ideally, in-house, we'd like to see an antibody with this signal intensity, but with the histogram allowed to be pulled back a bit. So in this case, dilution C, which would be 8 micrograms per mil, would be a little bit too high. Dilution A does not have the intensity that we're looking for, and so you may run into the problem of, if you increase the histogram on this, you could potentially increase any background that was previously hidden or run into additional errors. So for this particular CD20, we decided to go ahead and go with our 5 microgram per mil concentration, because when pulled back, this signal intensity matches that of dilution C. And you never want to use more antibody than is necessary, because you can preserve your money and you can preserve your overall time. Because if you have to order more antibody for your studies, say that you are running 20 different slides and you're running quite a high concentration, it does take time for ordering, and then you also run into the time of having to sit on your project and wait until that antibody comes in. You potentially may even have to revalidate if it's a different lot. So it's just best to use as minimal amount of antibody as possible with the output that you're looking for, the desired output. Once you have each of your antibodies at an optimal concentration, you can then initiate a full panel run. You can combine your antibodies and run the assay and see how it comes out. When you evaluate an image such as this one shown, the composite has about 8 or so antibodies here. You want to assess each channel just to QC and make sure nothing went wrong. There's always potential for error. And as we can see here, each of our channels is very specific to the antibody at hand, and there is minimal to no background observed. Especially here for our CD20 test case, we have just a few pockets of B cells, and that's what we would expect. Image QC and evaluation is always done, like I mentioned before, to evaluate any potential issues. So there could be signal loss, maybe some inefficient elution, tissue damage or loss, which is minimal but can still occur with the fluidics-based approach. There's a little bit of movement there. Imaging issues, potentially there's bubbling or maybe some focus issues. And then after all of this is completed, we prepare the data package and the raw images, and we can then transfer this to either the customer directly or to any analysis team. And with that, I'm going to pass it off to Mark, who is with one of those analysis teams.
All right. Well, thank you all for attending this webinar. I'm Mark Sadie, co-founder and chief technology officer of pathomics.io, a contract research organization specializing in end-to-end analysis of digital pathology datasets. So as a high-level overview, image analysis is the process of applying computer algorithms to process and interpret digital images of tissue samples in a quantitative manner. It's by no means a one-size-fits-all solution. At the start of your imaging study, it is important to consider the methods that may need to be developed to extract the results that you're looking for. And in this presentation, we will review a multiplexed image analysis workflow that we've developed and highlight some practical applications in several peer-reviewed studies. So multiplex immunohistochemistry is a technique that allows for the simultaneous detection and visualization of multiple targets, such as proteins, within tissue sections using distinct fluorescent labels. By enabling the analysis of multiple markers in the same tissue context, it provides a more comprehensive insight into the spatial relationships and interactions between different cell types or molecules. This method is somewhat particularly useful in research and diagnostic applications, such as studying tumor microenvironments or identifying complex immune responses, essentially enhancing our understanding of disease mechanisms and aiding in the development of targeted therapies. So it's easy to get overwhelmed at the volume of data generated from multiplex IHC studies, and attempting to visualize all markers simultaneously won't really yield any meaningful information as shown here. But by meticulously designing a multiplex workflow to profile specific combinations of markers and cell types, you can address and evaluate distinct spatial relationships between cells in their native spatial context. And some combinations can include proliferating tumor cells, activated T cells, macrophages associated with immune suppression, and regulatory T cells, to name a few. In this webinar, we will walk you through an example workflow from a recent project done in collaboration with the team at Fortis, and then show the practical application of such workflows from our published studies. First, we will review a multiplex analysis workflow developed for a recent project done in collaboration with Fortis, using open source tools such as QuPath for the image analysis, and Python for the downstream statistical analysis and data visualization. So, common to nearly all digital pathology workflows is cell segmentation. Cell segmentation is the process of digitally isolating and identifying individual cells within tissue images for detailed analysis. Historically, watershed segmentation has been used where the image is treated as a sort of topographic surface, simulating how water would fill different areas to find and outline each cell. Non-AI approaches such as watershed face considerable challenges in achieving accurate segmentations across all cell types. Variations in things like nuclear size, contrast to background, cell morphologies, all of these can impede the accuracy and reliability of this kind of segmentation. Advances in AI-based methods like Stardust outperform traditional techniques by using large training datasets to enhance segmentation precision. Stardust segmentation uses deep learning to accurately outline cells in crowded or complex images, handling overlapping and irregular shapes better than traditional methods such as watershed. There are multiple approaches to take towards phenotyping cells. Supervised classification is done in tandem with the guidance of a subject matter expert, such as a pathologist. Unsupervised approaches autonomously cluster cells to reveal distinct subpopulations and uncover new cellular insights without prior labeling. Using these approaches in tandem can actually maximize the insight derived from multiplex datasets, effectively balancing expert accuracy with algorithmic discovery. But in this presentation, we will focus primarily on supervised approaches and how this can be used to query specific cell types of interest. And the first step is thresholding our positive population, typically done in conjunction with a pathologist. Here, we showed this for several markers in our panel, such as CD8A, PCNA, and cytokeratin. So now that we've classified each cell for every marker, we need to combine these into a multiplex classifier. And by applying each of our pathologist-supervised classifiers sequentially, we can then get a list for each cell containing the markers that it is positive for. Cells can then be filtered from this to look at specific subsets of interest defined by more than one marker. We can identify cellular subsets such as regulatory T cells, activated T cells, proliferating malignant epithelial cells, and any other cell types definable by the markers included in the panel. Once defined, we can score tissues not only for single positive populations, but also the subsets defined by more than one markers of interest. In a fundamental sense, spatial biology involves techniques to simultaneously visualize and analyze multiple markers within their native spatial context. But up until now in this presentation, the spatial dimension hasn't really been utilized to its full potential. Area segmentation is one approach to analyze cells in relation to larger elements of the tissue microenvironment. Here, our tissue is annotated for representative tumor and stroma regions using cytokeratin. AI models are then custom trained to find these structures across all images in an experiment. We can then take our area segmentations and combine them with our classified and segmented cells. And this allows us to compute the distance of these cells to the nearest tumor stroma boundary. This can then be leveraged to identify subpopulations of cells immediately within regions such as a tumor mass as a means of characterizing phenomena such as immune infiltration. Some additional spatial features we can generate include cell distance analysis, which is the distance of each cell to the nearest PCNA positive cell, or Delaunay triangulation, which allows us to identify spatial neighborhoods. And while spatial analysis allows us to generate spatial features, what are the practical applications? So in collaboration with nephrologists at the Mayo Clinic, our aim was to use multiplex imaging to characterize the heterogeneity and identify biomarkers of kidney injury caused by immune checkpoint inhibitors. Immune checkpoint inhibitors are commonly used to treat a wide variety of cancers, but one major side effect is acute kidney injury hypothesized to be caused by the elevated systemic circulation of these immune cells. Prior studies have yielded a panel of potential biomarkers indicative of acute kidney injury caused by immune checkpoint inhibitors, and having the ability to characterize the immune landscape using these markers can actually identify the biomarkers relevant to differentiating acute kidney injury caused by immune checkpoint inhibitors relative to other causes, and fundamentally yielding a more promising prognosis for the patient. So as previously shown, we can segment our cells and begin visualizing marker expression patterns across different disease categories of interest. Independent of cell classifications, we can visualize marker intensity distributions grouped by the cause of kidney injury, termed code. Shown here is simply the mean CD3 intensity of all cells grouped by AKI code. And visually, it may appear as though there is no discernible difference, and that's because the intensities of the true negative cells typically outweigh those of the true positive cells, especially when viewing cell types commonly present in low abundances. However, by using our pathologist-supervised thresholding approach, we can isolate the positive population and score samples accordingly. So by applying the pathologist-supervised thresholds, we can then identify the positive population of cells, and find the percentage of cells positive for individual markers, as shown here for CD3. That being said, many immune cell types are characterized by more than one markers. As each cell in the dataset contains a list of markers that it is positive for, we can query specific immune cell subsets. Here, we measured the percentage of CD4 memory T cells as those that are defined as being CD3, CD4, and CD45RO triple positive. Tying this back to the original study, we scored our samples for a variety of immune cell subtypes to identify those that exhibit differential scoring for one AKI classification relative to others. And while many subtypes do exhibit variation, CD4 memory T cells, T helper cells, and dendritic cells were ones observed to be statistically significant. But as the old saying goes, seeing is believing. So it's important to validate that what our results show is consistent with the images themselves. Using additive color theory, we highlighted the relevant immune cell subtypes in each row using representative samples of AKI classifications as columns. In the top row, CD4 memory T cells were defined as those that are CD45RO, CD4, and CD3 triple positive. And given that each single positive cell type was labeled with a primary color, the triple positive population is shown in white. Similarly, T helper cells, shown in the middle row, were defined as CD4, CD3 double positive, and highlighted in cyan. And dendritic cells, defined as HLA-DR and CD11C double positive, were shown in yellow. So this pathologist-in-the-loop methodology for cell classification can be extended to additional studies that necessitate a comprehensive analysis of the spatial distribution of these immune cells. And on that note, let's discuss one application of spatial biology in characterizing the immune landscape of kidney transplant rejection. So more often than not, there is an underlying notion that once an individual receives a transplanted kidney, that they're on the road for recovery. In reality, many complications can arise. In fact, up to 15% of transplanted kidneys will exhibit signs of rejection during the first year of transplant. And identifying the cause of the rejection can actually enable clinicians to treat the rejection before it progresses and requires removal of diseased tissue. So in collaboration with the Mayo Clinic, we received 247 multiplexed images from 32 archived patient samples, bearing a confirmed diagnosis from one of 6 Banff classifications of renal transplant rejection. Our objective was to develop a method to characterize heterogeneity within and across patients and explore how the spatial dimension can yield a more accurate stratification. So in this study, we will demonstrate the pivotal roles played by each step of the analysis as previously outlined in this presentation. Just as previously shown, we can score samples grouped by a categorical variable such as disease classification. We can quantify the percentage of cells positive for an individual marker and visualize these results as a heat map. And while this does allow for the simultaneous visualization of several markers in tandem, we do lose a bit of information regarding the distribution of scores from samples across our population. With box plots, however, we can actually view individual samples as points and metrics of their distribution, such as median, interquartile range, and outliers. Here, we show the scores for CD4 memory T cells, again as being defined as CD3, CD4, and CD45RO triple positive. But at this stage, we've yet to really take into account any aspect of the spatial dimension, such as spatial localization of cell types to various renal structures. Here, we display the relevant markers for renal structure segmentation such that a pathologist can annotate their structures of interest. In this study, we were interested in the localization of various immune cells to glomeruli, tubules, arteries, and interstitial areas. And after obtaining over 3,100 annotations from our pathologist, we then trained a pixel classifier and applied it to all images in the dataset. Several different classifiers were used, and we varied the type of model, convolutions, feature scales, and markers used. But ultimately, we opted for the model with the highest overall accuracy in a blinded validation set. And much like how lymphocytes infiltrating into a tumor mass is more indicative of cancer patient prognoses than cell counts alone, the renal localization of immune cells can be indicative of rejection cause. We can apply the same percent positive scoring approach to explore differential localization of various immune markers and immune cell subtypes to glomeruli, arteries, interstitium, and tubules, with a representative image shown above and the structure highlighted in yellow. The main strength of this approach is that it guides cell type discrimination through the eyes of an expert observer. But we'd also like to investigate spatial features associated with the heterogeneity we've observed across our patients. So as part of this study, we have created a framework for predicting categorical variables from supplementary sample categories. And this isn't just limited to kidney transplant rejection categories. This can also be applied to any feature of a dataset in which comparisons across one or more groups is needed, such as differentiating a control from experimental treatment category. Once the model has been trained, we can identify the spatial features that played the most important role in classifying these patient samples. Much like we had done with the percent positive scoring heat maps, we can visualize the most available features for the classifier, grouped by rejection category. But as previously mentioned, heat maps lack the ability to portray population distributions, and when visualized as a violin plot, we can truly appreciate the heterogeneity in feature expression across disease types. This indicates that it's not always just one feature actively differentiating a disease, but rather multiple in tandem. But again, as the old saying goes, seeing is believing. So here, we randomly sampled 40 regions across our samples with the raw EC-adherent channel shown along the DNA counterstain. Next, we show an overlay of a non-spatial feature, in this case, mean EC-adherent intensity, across a whole cell. And you can see that there is some variation across our samples. But now, if we visualize the fifth most important feature in our model, a greater degree of inter- and intra-sample heterogeneity can be seen. But how does this fare in practice when trying to resolve two similar disease categories? Well, historically, chronic and acute antibody-mediated rejection have been quite a challenge to differentiate. And while variable expression of EC-adherent has been observed in the past, the difference here is quite minor. But when viewing our fifth most important feature, there is quite a stark contrast in feature expression. That being said, this is just one of the many features working in tandem to differentiate chronic from acute antibody-mediated rejection, as well as other disease subtypes. And the takeaway message here is that analysis tools that incorporate spatial proximity and localization are capable of revealing novel forms of intra- and intra-microenvironmental heterogeneity. So in short, we have introduced an open-source pathologist-in-the-loop workflow for the quantitative assessment of microenvironmental heterogeneity using the spatial domain. We have demonstrated the applicability to a wide range of multiplex studies and support across multiple imaging modalities. And finally, we highlighted several publications from past customers ranging from cancer immunotherapy, preclinical drug evaluation, and multiomic characterization of tissue microenvironments. So on that note, I'd like to thank everyone shown here, but primarily the Fortis team, not only for coordinating this webinar, but for also generating some of the highest quality multiplex images I've ever had the privilege of analyzing. There's an old saying which we typically use when dealing with poor quality images with, let's say, overly ambitious analysis goals. Garbage in, garbage out. But working with Fortis, I have to say my experience is more like treasure in, treasure out. I am confident that the results that we have provided to our customers are truly reflective of the biological underpinnings of their dataset, and I hope that these results will further enhance their understanding of their patient spatial biology and continue to be of further value to future customers, hopefully such as yourselves. And on that note, I'd be happy to take any questions, comments, and hopefully not, concerns. Thank you.
Alrighty. Thank you so much for that presentation, Danielle and Mark. I hope our audience found it of great interest and value. If you do have any questions for our speakers, then please continue to submit them using the Q&A button at the bottom of your screen. Unfortunately, Mark won't be joining us for this Q&A session, but we have CEO and co-founder of Pathomics.io, Trevor McKee, here to answer any questions you may have about Mark's part of the presentation. Thank you for joining us, Trevor. We'll now address some of our audience questions. So I think a few have come in for Danielle. What current services do you offer?
Currently here at Fortis Life Sciences, we offer anything ranging from the actual tissue preparation all the way to the imaging, staining and imaging for both the COMET and the PhenoImager HT.
Excellent. And then again for Danielle, what are some of the most common problems that arise with multiplex staining?
I would say the most common is just primary antibodies not working. It's very important that you source your antibodies and then bring them in-house to optimize extensively before you actually apply them in a panel. Secondary to that, I would say improper tissue fixation and even inefficient elution.
Thank you. And then what technology were the renal studies performed on, and what size was the panel?
Thanks for that question, Emily. So there were 27 markers in the panel, and that was an imaging mass cytometry study. But we've worked on a number of different modalities, including IMC and COMET and other multiplex tools as well.
Excellent. And then another question for you, Trevor. Who owns the IP generated from a study?
Oh, sure. Great question. So any results arising from the analysis, of course, resides with the person, the customer who requested the work. And we tend to only ask that any methods that we generate over the course of performing a study, we have the right to reuse those methods for other projects as well.
Sure. And then Mark mentioned unsupervised clustering briefly. Could you elaborate a little bit on that?
Sure. Yeah. So there's, I think he mentioned the supervised approach is where you use pathologist guided thresholds. Unsupervised clustering is just letting the data cluster according to similarities in expression pattern between the cells. And so with unsupervised clustering, you can get outputs that indicate whether things are more biologically similar or different based off of the expression patterns present within the tissue.
Brilliant. And then Danielle, how big of a role does tissue fixation play in the output of an assay?
Yeah, I would say it plays a huge role. A lot of times, if it's improperly fixed, it can cause issues with autofluorescence, which is one of the bigger issues that we observe. And occasionally, if it's improperly fixed, it can even experience some damage if it goes through Kehr cycles with cyclic IF. So it's very important that you really take the time and fix it properly.
Mm-hmm. Excellent. And then you mentioned data sharing. What platform are you using to share images with customers?
Currently, we use PathCore, and so we can upload those images fairly quickly, and then they're readily available to the researcher.
Excellent. And then Trevor, we've got a question about the use of AI in your analysis. Can you talk a little bit about that?
Certainly, yeah. So there's a number of places where AI comes in. The first is during the segmentation, as Mark mentioned. We're using pre-trained AI models that are trained off of manual annotations of many different cells, and what that allows is for a better segmentation using these AI computer vision based methods than is possible with sort of more classical machine learning or watershed and thresholding. The other place where AI comes in is, as mentioned, in the post-processing analytics. So we've built a machine learning classifier to predict the cause of transplant rejection was the one thing we showed there, and we can build classifiers that can learn from the data to make certain predictions or correlate with outcome as well.
Brilliant. And then how can our audience members send you their images?
So we're very flexible. We can either, if they already have an existing Dropbox that they can share with us. We're happy to do that. As Danielle mentioned, there's PathCore available within Fortis. That can also be a method to transfer files, and we can also provide a Dropbox, cloud-based Dropbox, where people could upload their images as well for us to analyze. But we're very flexible to work within the confines of whatever the institutional requirements are on the other end.
Excellent. So then that is kind of goes, I think, hand in hand a little bit. How large of a data set can you analyze?
I don't think we've received I don't think we've found the upper limit. We've worked on things up to like some of the spatial transcriptomics data sets are up to a terabyte in size. I think larger images will take longer time to process, but in general, there's no challenge with analyzing large images. Yeah.
Brilliant. And then Danielle, is the characterization step that you talked about really necessary for imaging on the comet?
Yeah, I would definitely say that it's absolutely necessary, because you always want to optimize your antibodies first in whichever way your lab typically does it. But when you actually take the antibody and you translate it onto the comet, you want to make sure you have at minimum 3 different sort of dilutions at different ranges, like I mentioned, so that you can observe any drop off in signal, and then you actually get the optimal working concentration for your studies on either your own personal tissue or customer tissue in this case.
Okay, and then for a typical project, what would the turnaround time be?
That, I would say, varies on the number of samples, and also whether, being that we're an antibody company ourselves, if you use the antibodies in-house, it would go a lot quicker, because then we're eliminating that optimization step. But I would say a good timeline for the COMET project is often 10 to 16 weeks, really depending upon that source of antibodies. And then for the PhenoImager, it's a bit quicker, and I would say on the safe side, 6 to 8 weeks.
Okay, brilliant. Thank you. And then Trevor, I think you did touch on this a little bit, but what kinds of imaging modalities do you support, and what tools do you use for the analysis?
Great question. So we've had a rather wide experience across a wide range of modalities. I think the exact ones are listed on our website, but we've done anything from mass spec based work of imaging mass cytometry, or even MALDI metabolomics data sets, through fluorescence based, whether that's the PhenoCycler, the PhenoImager, the COMET, and then just sort of traditional immunohistochemistry as well, and then we do also offer the ability to analyze spatial transcriptomics data, either in parallel with the multiplex fluorescence, or by itself as well.
Excellent. And then will the platform be deployed in the cloud?
So we're definitely looking at a cloud-based procedure for performing the analysis. Right now, the portion that's in the cloud would be the PathCore. It allows you to be able to view the images that are generated from Fortis staining in the cloud, and be able to zoom in and out and pan around. We are looking at ways that we can incorporate what we're doing with PathCore and with other cloud-based platforms, as well as developing our own to be able to provide that interactive ability. For right now, what we do is we do the analysis and provide results back to the client directly.
Brilliant. Well, thank you very much, Danielle and Trevor, for answering our audience questions today. Before we wrap up, is there any last comments you want to share?
I might just say that, however you choose to do your staining and analysis, I think it's important to make sure that you you think through the entire process from, from panel design through to the end, because sometimes, for example, there might be a structural marker you might want to include, that if you designed it into the panel, it makes your analysis easier because you can use that marker to discriminate, let's say, tumor from stroma, these sorts of considerations. So it's definitely useful to have kind of an end-to-end approach when you're doing this sort of work. And we've benefited from working very closely with the Fortis team to make sure that it's a seamless transition from the imaging portion to the analysis.
Excellent. Okay, well, I would just like once again to thank our wonderful presenters for being giving an excellent presentation and answering our audience questions. Thank you, Trevor, for stepping in. And I would also like to thank our audience for your time and questions. Don't forget to visit us at biotechniques.com for more webinars. And thank you again for attending. Goodbye.